Okay. Now, Anand, right? Anand Sarma.
Yes, sir.
Okay. Are you okay and feeling comfortable?
Yeah, sure, sir.
Okay. So, Anand, just introduce yourself in a brief so we can start further.
Sir, I am Anand Sharma. I come from district Morena of Madhya Pradesh, and I have done my graduation from IIT Guwahati in biotechnology and post graduation from IIT Madras in bioprocessing engineering. And sir, also I have done a research internship from Seoul, South Korea in the field of nanotechnology. And sir, my interests are playing keyboard and keeping my body physically fit and well.
Okay. Anand, as you mentioned in your form, that is, you are keen interested in the immunology, bioprocess engineering, or fermentation technology. Okay.
Yes, sir.
So, my questions to you: how you can define and how you can differentiate in bioreactor and fermenter? What is the difference in bioreactor and fermenter?
Sir, bio, as far as I know that fermenter is used for if any microorganism or enzyme is involved process, then that process is known as fermentation, and that if that bioreactor is known as fermenter. But if any other process is involved of in containing biological process, then we will call it bioreactor. So this is my basic understanding.
Okay, Anand, tell me why there is a need of a fermentation engineering or fermentation technology in related to the biotechnology. What is the applications of fermentation technology or bioprocess engineering for human reference?
Sir, there are a lot of applications of fermentation technology.
Yeah.
For example, production of pharmaceuticals, antibiotics are there, and then production of vitamins, which are essential for the human growth. And then, sir, production of beverages are there, biofuels is there.
Okay. I think network issue is from your
Human.
side.
Yes, sir.
Okay, okay. As you stated that pharmaceutical product for the production of pharmaceutical as well as biofuels, etc. Okay,
Yes, sir.
okay. Anand,
Sir, can you please
for the production
repeat
of
it, sir?
a pharmaceutical compounds or pharmaceutically important molecules, we need DNA. That we need RDT technology. That means recombinant DNA technology. Okay.
Yes, sir.
So, can you justify that genetically modified process or transgenic process is important for the productions of a pharmaceutical compound at the level of industrial? How RD technology is related to the production of a pharmaceutical compounds at the industrial level, production of at the industrial level?
Sir, generally we need pharmaceuticals for human consumption. So, and sir, human cells are very difficult to grow in fermentation technology. So, the gene of interest, which is responsible for producing those pharmaceuticals, are inserted into the microorganisms, and then which helps in producing those proteins which are required or vitamins for the human consumption. So without RDT, it is not possible to produce uh, human proteins which are essential for our growth. So this
Okay.
is
Anand,
uh,
can you suggest some examples of pharmaceutically important compounds which have been developed by using RDT technology or transgenic techniques? Can you name some pharmaceutically important compound?
yes, sir. I can remember right now that the insulin is generated from fermentation technology, for which is used in diabetic patient to cure diabetic patient. So that is one, and then sir, producing growth hormones and growth factors using this RD technology. We can use fermentation technologies.
Tell me something about insulin. What is insulin?
So this is a uh, hormone which generated inside the body to regulate the blood sugar level. And in diabetic peoples, the blood sugar level increases. So to control that, we need to give an external dose of insulin, and so we produce through by fermentation technology that insulin, and we give to the diabetic patients.
Okay. Anand, have you heard about the green biotechnology or blue biotechnology?
Yes, sir.
Yeah. What is the meaning of a green and blue
Sir,
biotechnology? Yeah.
sir, when the blue biotechnology means when the marine resources are involved in production of uh, various biotechnical products, then we call it blue biotechnology. And if in green biotechnology, we produce like which are safer alternative to the pollution and pollution free or lesser pollution content are there. Then if we are using biotechnology in that field, then we call it greener biotechnology. For
Okay.
example, biofuels are there.
Okay, okay, okay. Generally, we consider genetically modified are good for human health or human welfare, right? So, being good for human welfare or human health or also human society, genetically modified organisms are not allowed to be cultivated in modified plants are not allowed to cultivate on a large scale by the government. Why there is a ban on the cultivation of a genetically modified plant at the level of agriculture level?
Sir, there is ethical concerns with genetically modified organisms that we do not understand fully that how these microorganisms will behave in future, because generally biological processes are long processes, so they require uh, uh, long-term studies to check out whether how it will affect your body. And so those studies, we are we are very new in this field, and we do not know how it will impact the human body. So it is considered as unnatural process which can have adverse effect on human body. For example, it may create allergy, and it can create many other problems inside the body. So because of that, government is not allowing it.
Anand, can plant material or plant part can be used as a vaccine in the future or right now? Have you heard about the edible vaccine?
Yes, sir.
Yeah.
There is
What
a
is
process called edible vaccine in which, for example, the plant products like potato and sugar beet can be used as a vaccine. So you can just eat it, and it will act as a vaccine. So there is a possibility in future.
Sorry, I was unable to listen you properly because there was a network issue. Can you repeat your answer once again?
Yes, sir. I was saying that yes, there is a possibility of edible vaccine, which is going. That it is under the research is under. It is going in the right direction, and few potential candidates are like potatoes and sugar beets are there, which can be used as edible vaccine.
Okay. Can you suggest some name of a genetically modified plant?
Yes, sir. BT cotton is one which we are doing in India, and sir, BT brinjal is there, BT mustard is there. So these are few genetically modified plants.
Okay. So in fermentation technology, we uses fermenter for the production of any desirable molecule at the level of industrial, right? So,
Yes, sir.
what are the possible parameters to construct a fermenter to design a fermenter? What will be the parameters to be considered for the designing of a fermenter or bioreactor?
Sir, the main concerns with bioreactors are that we need a good productivity and at a sustainable way. So to maintain that productivity and efficiency, we need to control a lot of factors which are involved in biological processes. For example, pH is there, oxygen concentrations are there, and the temperature and media composition and the substrate and all. We need to consider those factors while producing any uh, product from fermented technology. So these conditions should be optimized to get the optimum yield. So these are the main constant in the uh, fermenter step of fermentation technology.
Okay. Anand, as you are a postgraduate with environmental biotechnology, right?
Yes, sir.
So, what is the bioremediation?
Sir, bioremediation is a way to degrade the pollutants with the help of microorganism, and in which the heavy metals, the various kind of pollutants are there. So different kind of microorganism can be used to degrade that, and so that the pollution should not be there.
Okay, nowadays abiotic stress is one of the major factor for the plant and as well as other animals growth and behavior, right? So many abiotic stress like as salinity, drought, heavy metal toxicity, etc. In your environmental biotechnology, if suppose you are a research scholar and you are doing some kind of a research on your laboratory and you wanted to remove the abiotic stresses from a particular kind of a area to mitigate the negative impact of that stress for the plant and organism, right? So what kind of approaches you would apply to remove or to mitigate the abiotic stresses from a particular kind of area?
Sir, uh, we can provide artificial ways to create the oxygen conditions there.
In simple,
And
sorry, in simple way, my question is that the being environmental biotechnology, how you can revoke or how you can mitigate the abiotic stress of the plant by using environmental
yes,
biotechnology approaches. Yeah.
sir, we can one method which I can suggest, that we can use some microorganisms which generate oxygen levels, and we can spread in that area, so which will improve the oxygen concentration in that area.
But what is the relation of oxygen concentrations in terms of abiotic stress like as a salinity, drought? Suppose a plant is suffering from salinity, right?
Yes, sir.
Salinity is one of the major detrimental factor of abiotic factor, which affect a lot of on the plant growth and development. Suppose the plant is suffering from salinity impact, salinity stress,
Yes,
then how oxygen concentration will minimize or reduce that salinity stress?
sir, in that case, we can have some GMO crop which can transfer its gene to these plants with the cross pollination, so that the salt tolerance should come in those plants. This can be one way, so that in that area we can have some GM crops which will in future, which cross pollination will happen in those plants, and the salt tolerance capacity will come in those plants. So in that way, they can, tolerate those salt concentrations and can survive.
Okay, Anand, you stated that sustainability, a word you use sustainability, right? What is
Yes,
the
sir.
meaning of sustainability?
So sustainability means that you are using resources without compromising with the future generations. The resources which your future generation is going to come, they should not suffer because what you are doing. So that is the sustainable way of living.
Okay, uh, very good, well done. Anand, you are interested in the immunology, right? Your favorite
Yes, yes, sir.
area of interest is the immunology. So, can you suggest some techniques to find out the antigen antibody interactions? Suppose you are in a pathology laboratory and you have some sample of blood, and you want to check out
Yes.
the interaction of an antigen and antibody. So which kind of a technique you will apply to identify the interaction of a protein and antigen and antibody? Can you name some techniques?
Yes.
Yeah.
So there are a lot of techniques which I can remember right now. Is one is ELISA test is there, in which the antibody interaction can be found out with the help of detection of the intensity of light. And
Hello, Anand. I think network issue is there. Anand, yeah, you can continue.
so am I,
Yeah, yeah. Right
so am
now
I
you
visible?
are visible and audible also, so you can continue with your answer.
Yes, sir. So, sir, I was talking about. So can you help me?
Yeah, you were answering some your answer.
Yes, sir. I was talking about the and one of now.
Anand, I think network issue is so severe towards your, so I am not able to listen you properly because after just few minutes or few seconds, your video has gone to be paused. Hello,
Hello.
hello.
Hello. Yes, sir. Am I audible?
Yeah, but you are you are showing here in the two pictures. Have you logged with another account?
No, sir. Let me. Sir, can I join again?
Yeah, it will be better.
So now,
Yeah, now you are visible in a single frame.
sir, I don't know what is happening today.
Actually, it's a matter of a network, and I think from your side there will be a severe issue of network. That is why your voice is not coming properly, and your also video is not coming properly. But don't worry. If you're not comfortable with this network, we can have a phoning or calling interview. I will ask whatever I want to ask here. So it will be better. If you feel comfortable in phoning, then I will also conduct a phoning interview. Okay. So if
Yes, sir.
yeah, so we will continue.
So right now it's okay.
Yeah,
Yes, sir. Yes,
yeah.
right,
Right
sir.
now it's okay. You can just proceed.
Yes, sir.
And the last question was what?
Sir, I was talking about the techniques which are involved
Yeah,
in
exactly.
antibody antibody.
My question was that uh, suggest some technique to identify to detect the antigen antibody interaction.
Yes, sir. So I told that one is ELISA and another one was fluorescent that affects system effect
Okay.
technique is there.
Anand, have you heard about the RIA?
Yes, sir. I have heard about it.
Yeah. What is this? What is RIA? RIA stand for what, and what is the principle of RIA?
Uh, Sorry, sir. I'm not able to recall right now.
Okay, no issue, no issue. Don't worry. Uh, you don't have idea about the RIA.
Yes. I have to
So,
read about it, sir.
okay. So, come to the next point. In immunology, we often use Western blotting.
Yes, sir.
Have you heard about it?
Yes,
What
sir.
is the Western blotting and what is the working principle of Western blotting?
So Western blotting is a way to identify a particular protein. Uh, so the principle behind is that the probe is generated against a protein, and that probe is it get stuck on the one surface. Then it will be visualized with the help of radiography, so that we can find that particular interest of part of the protein which we are interested in. So this is the technique involved in this process is known as Western blotting.
Okay, Anand, there are two terms, that is agglutination and precipitation.
Yes, sir.
How you can distinguish between these two terms, agglutination and precipitation? Is there any differences or both are the same?
So definitely there is a difference, and sir, in agglutination, sir, the smaller particles are involved, which with the help of the antibody interactions, they can be bring down in the system, while the precipitation happens of the microorganisms with the help of antibodies. So in agglutination, the smaller particles are involved, antigens, while in precipitations, the bigger microorganisms are involved. So this is the main difference which I can call right now.
What is hybridoma technology?
Sir, hybridoma technology is a method to generate monoclonal antibodies. And sir, in this, the cancerous
Okay,
myeloma
need
cells.
not to be go for
So,
the mechanism.
yes,
You
sir.
said, that is hybridoma technology is a technique to produces monoclonal antibody. What is
Yes, sir.
monoclonal antibody?
So monoclonal antibodies are very specific antibodies to any antigen. So they can interact with only one antigen at a time. So very specificity is there in those antibodies.
Then what is polyclonal antibody?
So polyclonal antibodies can bind with multiple epitopes at a time of an antigen.
What is
So that
paratope?
the paratope is a region on the antibody which interacts with the antigen.
And what is epitope?
So the epitope is the surface on the antigen which interacts with the antibody. So that region is called epitope, and paratope is on antibody. That region that interacts with the antigen, that is paratope.
Okay. Uh, Generally, we define antibody are the glycoprotein, right?
Yes, sir.
For the definition of antibody, we define antibodies are the glycoprotein.
Yes, sir.
Is there any significance of a glyco term or glycosylation of a protein molecule for the antibody? Why antibody cannot be simple protein? It is a combination of a the glycoprotein. What is the significance of glycoprotein?
Sir, in the complement system, the glyco part, that lectin part plays a very important role in lectin pathway. So that interaction to happen, that glyco part should be there in antibody. So because of that, it plays an important role in clearance of the antigens from the system with the help of that lectin pathway. So this is one significance which I can remember right now.
Nature of a antibody is either soluble or insoluble.
Sir, it is soluble in
Soluble
blood
in blood.
plasma.
So,
Yes, sir.
what the factor is responsible for solubility of that antibody in the blood?
Sir, because the nature of the blood is polar in nature, so if the COO minus and NHC plus groups are there in antibodies, these plays an important role. These provides the polarity to the antibodies, which helps in solubilizing in blood. At the end, antibody gets solubilized in blood because of this polarity of antibody.
Okay. For the solubility of antibody, is there any role of a glyco part of the glycoprotein of antibody? can we say that glycosylation, yeah, glycoprotein responsible for the solubility of our antibody?
So it may be, I am not aware. Sorry, sir.
Okay, not no issue. uh, Anand, tell me one thing. Antibody are very specific for a particular kind of antigen, just like a
Yes,
enzyme and substrate, just like hormones and receptors.
yes, sir.
So, is there any similarities in the interaction of antigen, antibody, hormones, receptors, and substrate and enzyme? Pattern of binding of these three different kind of molecules. Is there any similarities, or they are totally different from each one?
So definitely there must be similarity because the ultimate interaction which is happening between the molecules is based on the chemical interaction, which is hydrophobic interaction and yeah, ionic interactions are there, and which are because of the same kind of bonds are present on these molecules. So the interaction happening is similar kind of interaction only. It is just that what kind of bond is forming and what is the shape and size which is recognizing that differentiate the different kind of signals.
Okay, what different kinds of possible chemical bonds are involved in the antibody structure?
So covalent bonds are there, and so disulfide bonds are there, and so ionic interactions are happening, and so hydrophobic interactions are there. So these are the major interactions which are happening, one over interactions, I would say.
Okay, for the formation of a disulfide bond, which kind of amino acid must be present in that protein?
Sir, so the proteins which contain sulfur groups such as cysteine, uh, it must be there so that the disulfide will be formed.
Have you heard about the Hing region of the antibody?
Yes, sir.
Hinge, Hinge region.
Yes, sir.
What is
Yes,
what
sir.
is this?
So this is the region which connects the heavy regions with the heavy variable regions with the uh, so that light chain and heavy chains are connected through this region. So that's called the hinge region with the help of disulfide bond. Two disulfide bonds are there.
Okay, any kind of antibody have has two different measure two role, that is effector role and antigen binding role. Okay, so which part
Yes,
of
sir.
antibody is responsible for the effector function of that antibody?
So the heavy chain, the lower heavy chains are used as a factor. I
There is
mean,
a
they have
specific term
factor
used for
function.
that. What is known as?
So FC region is there.
Very good. What is the FC?
So sorry, sir, I'm not able to recall.
Another also term is a FAB. These two
Yes, sir.
fragments are known as the FAB and FC. So what is stand for FAB and FC?
Sir, C for constant, I'm sure, and AB is for antibody, antigen antibodies, as
C
far as
for
I can know.
crystallizable or not constant. FC का मतलब
Okay.
होता है, SB means that is what? Fragment of crystallization.
Crystalline.
And
Okay, sir.
FAB का मतलब होता है fragment of antibody और antigen
Antibody.
binding.
Antigen. Yes, sir.
ठीक है. So
Thank you, sir.
okay. ओके आनंद, आपके पास कुछ आपके मन में कुछ सवाल है जो कि इंटरव्यू में जाने से पहले आप पूछना चाहते हैं?
Yes, sir. I have one question regarding area of interest.
या
Because sir, my project was on on computational biotechnology, and I have done a research internship in the field of nanotechnology. That computational thing also contains nanostructures. I was characterizing those nanostructures. So there was a KFFE tetamer was there. So with the help of that, I was trying to analyze that what kind of nanostructure it will form through computational biotechnology. So, sir,
सो
should I keep nanobiotechnology as my area of interest? And
Okay,
then fermentation
just a
technology
minute.
and engineering.
Currently, what are you doing, Anand?
Sir, I am preparing for UPSC civil service.
UPSC. It's okay.
Yes.
So at the level of a master, your master, which subject you have more confident? That is immunology, biochemistry, molecular biology, nanobiotechnology, whatever.
So these types of subjects was not there in masters.
Okay.
My master was on bioprocessing,
Yeah.
so most of the subjects was on thermodynamics was there. Then bio fermentation technology, bioreactor design,
Okay.
bioprocess
So
control, all the.
okay. Got it. So among all of them subject, all of them paper, which paper you would prefer to be more confident?
So I will go with fermentation.
Fermentation.
I am Yes, sir.
Okay, so definitely, suppose interviewer will ask to you what is your area of interest and what is your favorite subject, then definitely you would say that is fermentation technology. And what you have to do, just search and just find out the some technical term related to the fermentation technology. That is bioprocess engineering. मीन्स
Yes, sir.
व्हाट सपोज अगर आप इस बात को स्टेट करते हैं दैट इज योर एरिया ऑफ इंटरेस्ट इज द फर्मेंटेशन टेक्नोलॉजी और बायो प्रोसेस इंजीनियरिंग दैट मीन्स someone is expecting to you that you have to answer all latest terms of the fermentation technology or bioprocess engineering, right? And definitely there will be a such kind of a question that what is your favorite topic or what is your favorite subject and what is your interest of area. Such kind of a questions will definitely be there. ओके एंड ऐसे ही क्वेश्चंस का आंसर देने के लिए आपको उस टॉपिक पे थोड़ा ज़्यादा कॉन्फिडेंट होना पड़ेगा। जैसा कि अभी आपने बताया कि आपको इम्यूनोलॉजी पे भी कॉन्फिडेंट है और फर्मेंटेशन टेक्नोलॉजी पे भी कॉन्फिडेंट है, राइट? तो
Yes, sir.
आपको आपको जिसमें जो लगता है ज़्यादा बेटर लगता है, इधर फर्मेंटेशन टेक्नोलॉजी या फिर आपका दैट इज़ इम्यूनोलॉजी, जिस किसी पे भी आपको ज़्यादा कॉन्फिडेंस लगता हो, जैसा भी आपने जो आंसर्स किए हैं वो भी उस पे भी हम लोग डिस्कस करेंगे। अब आपका टर्न
Yes,
हो
sir.
गया तो हम आपको सजेस्ट करेंगे क्या क्या चीज़ें करनी है। उन दोनों में से जो भी आपको लगता है आप उसको अच्छे से थोड़ा देख लीजिए। आपका इंटरव्यू कब है? दो को ही?
So third go.
थर्ड को है। अच्छा, ओके, तो
Yes.
आपके पास अभी एक दिन का लेफ्ट टाइम है। अभी
Yeah.
आप करेंटली कहाँ हैं? न्यू दिल्ली में?
Yes, sir.
ओके, तो आप अगर इम्यूनोलॉजी को अगर आप बोलना, आपने मास्टर्स में मेरे ख्याल से इम्यूनोलॉजी नहीं पढ़ा है।
No, sir.
ओके, तो आप
बीटेक
इम्यूनोलॉजी,
में पढ़ा है सर।
हाँ,
बीटेक में पढ़ा है सर।
ओके, तो आप इम्यूनोलॉजी को छोड़ दीजिए।
अच्छा, ओके
ठीक
सर।
है, बेटर होगा। आपने जो मास्टर्स में अपने एजुकेशन लिया हुआ है उनमें से जैसे सपोज आपने बोला कि फर्मेंटेशन टेक्नोलॉजी का, तो आप फर्मेंटेशन
Yes.
टेक्नोलॉजी पे थोड़ी सी अच्छी अपनी पकड़ बना लीजिए। जैसे बायो रिएक्टर और फर्मेंटर में क्या डिफरेंसेस है, कितने टाइप के फर्मेंटर्स होते हैं, राइट? कौन कौन से फर्मेंटर का किस किस टाइप के प्रोडक्ट का यूज़ करने के लिए यूज़ किया सकता है, एक फर्मेंटर को डिज़ाइन करने के लिए किन किन एस्पेक्ट्स का ध्यान जाना चाहिए, फर्मेंटर को ऑपरेट करने के लिए जो सूटेबल कंडीशंस होते हैं, टेम्परेचर, पीएच एक्सेट्रा, ये सब क्या होना चाहिए, फर्मेंटर को डिज़ाइन करने के लिए किस टाइप के वेसल्स का यूज़ किया जा सकता है, राइट? ये सारे जो चीज़ें हैं। और फर्मेंटेशन टेक्नोलॉजी का जो एप्लीकेशन है वो बहुत ब्रॉड है
Yes,
ना?
sir.
किसी भी चीज़ की जो प्रोडक्टिविटी है, जिसकी सप्लाई बहुत ज़्यादा अमाउंट में है, उसको करने के लिए फर्मेंटेशन टेक्नोलॉजी के थ्रू होकर के जाना पड़ता है। जैसे सपोज
Yes.
इंसुलिन की रिक्वायरमेंट बहुत ज़्यादा है, सो मेनी नंबर ऑफ पेशेंट्स आर देयर ऑफ डायबिटीज, सो उसका
Yes.
प्रोडक्शन कराने के लिए आपको फर्मेंटेशन प्रोसेस से होकर गुजरना पड़ता है, मतलब उधर की तरफ जाना पड़ता है। तो ऐसे कुछ इंपॉर्टेंट जो आपके फार्मास्यूटिकली इंपॉर्टेंट केमिकल हैं, कंपाउंड्स हैं, मॉलिक्यूल है, जैसे आपने बताया इंसुलिन, तो इंसुलिन को लेकर के थोड़ा आप कॉन्फिडेंट हो जाइए कि इंसुलिन क्या है। एक हार्मोन्स है जो कि आपका पैंक्रियाज से सेक्रेट आउट होता है। इसका रोल है हमारी बॉडी में ग्लूकोज के इंटेक को क्या करना, मिनिमाइज कर देना और
Very good.
ग्लूकोज के लेवल
Yes.
को मिनिमाइज कर देना, राइट? इसके पॉसिबल कॉन्सिक्वेंसेस क्या हो सकते हैं, डायबिटीज मेलाइटस का क्या कॉन्सेप्ट है, थोड़ा इन टॉपिक को आप अच्छे से पढ़ लीजिए। ग्रोथ जो फैक्टर आपने बताया, उस ग्रोथ फैक्टर का क्या एप्लीकेशन है उसको आप देख लीजिए थोड़ा सा। तो जो भी फार्मास्यूटिकली कंपाउंड्स हैं इनका इम्पोर्टेंट है, जो कि आपके से इंडस्ट्रियल लेवल पे प्रोडक्शन कराने के लिए फर्मेंटेशन टेक्नोलॉजी से गुजरना पड़ता है। और दूसरा, बेवरेजेस पे थोड़ा काम कर लीजिए। जैसे बहुत सारे बेवरेजेस इनका प्रोडक्शन फर्मेंटेशन टेक्नोलॉजी में किया जाता है। आपने एरिया
Yes.
बताया है कि आपका फर्मेंटेशन टेक्नोलॉजी है, तो डेफिनेटली देयर विल बी अ सच काइंड ऑफ क्वेश्चंस। बेवरेज प्रोडक्शन कराने के लिए कौन कौन से माइक्रोब्स को यूज़ किया सकता है, बियर प्रोडक्शन कराने के लिए क्या कर सकते हैं, वाइन प्रोडक्शन कराने के लिए क्या कर सकते हैं, राइट? तो ये थोड़े ऐसे कुछ एरिया होंगे जिनको कि आपको पढ़ना रहेगा। तो अभी आज का टाइम है और कल का टाइम है, परसों आपको जाना है।
Yes,
तो
sir.
फर्मेंटेशन टेक्नोलॉजी पे या फर्मेंटेशन वाला जो कॉन्सेप्ट है उसको थोड़ा अच्छे से, ज़्यादा डेप्थ में ज़रूरत नहीं है, लेकिन जो टर्म्स हैं उनको थोड़ा आप समझ जाइए। जैसे वैसे आपने आंसर्स, काफ़ी सारे क्वेश्चंस का आंसर बहुत सही किया, जैसे बायो रेमिडिएशन आपका आंसर बहुत अच्छा था, और हाइब्रिडोमा टेक्नोलॉजी का आंसर आपका काफ़ी अच्छा था। तो ये सारे जो आंसर्स आपने दिए वो ठीक हैं, ऐसे ही आंसर की रिक्वायरमेंट होती है वहाँ पे। और कुछ
Yes.
आपसे इस तरीके के भी सवाल पूछे जा सकते हैं कि इसका क्या पॉसिबल कॉन्सिक्वेंसेस हो सकता है। सपोज आज के डेट में इंडस्ट्रियल स्केल पे प्रोडक्शंस कराने के लिए फर्मेंटेशन इंडस्ट्री बहुत ज़्यादा बूम कर रही है, राइट?
Yes, sir.
फर्मेंटेशन इंडस्ट्री में अगर कुछ प्रॉब्लम कैलेमिटीज़ आ जाती हैं, कोई दिक्कत अगर आ जाती है, तो उसका कंट्री के इकोनॉमी पे क्या प्रभाव पड़ेगा या फिर पब्लिक हेल्थ पे क्या इम्पैक्ट पड़ेगा, ये थोड़ा-सा आपको सोचना पड़ेगा। इसको सोच करके आंसर आप उसको कर सकते हैं। तो थोड़ा-सा जो भी आपके एस्पेक्ट्स हैं, उनको पब्लिक वेलफेयर के कंसर्न में रिलेट कर लीजिए, क्योंकि
Yes,
अभी
sir.
करंट में यही चल रहा है और अल्टीमेटली जो गोल है वो यही है, ह्यूमन वेलफेयर के लिए सारा काम करना। बायोटेक्नोलॉजी का मेन पर्पज़ यही होता है ना?
Yes.
तो 2-3 काम आपको करना है। बाकी आपका बोलने का तरीका ठीक है, कोई प्रॉब्लम नहीं है। ड्रेसिंग आप बहुत स्मार्ट लग रहे हैं डेफ़िनिटली, उसमें कोई
Thank you, sir.
उसमें कोई दिक्कत नहीं है। और आवाज़ भी आपकी ठीक है, उसमें प्रॉब्लम नहीं है। इंट्रोडक्शन भी आपने ठीक से दिया है। इंट्रोडक्शन में अगर हो सके तो एक चीज़ को वहाँ से कट आउट कर दीजिए। जैसे आपने बताया कि आप मास्टर्स डिग्री के बाद आपने इंटर्नशिप किया और उसके बाद आप ये कर रहे हैं। मास्टर डिग्री का पार्ट होता है डिसर्टेशन, उसको अलग से स्पेसिफ़ाई करने की ज़रूरत नहीं है कि आपने इंटर्नशिप किया हुआ है। अगर वो पूछते हैं देन आप उससे स्पेसिफ़ाई कर दीजिएगा, ठीक है?
Okay, sir. Okay, sir.
सिंपली आप अपना बताइए कि आपने यूजी, पीजी कहाँ से, कौन से ईयर में किया हुआ है कंप्लीटली, और करंटली आप क्या कर रहे हैं, ये बताना भी आपके लिए थोड़ा ठीक रहेगा। करंटली आप तैयारी कर रहे हैं,
Yes, sir. So
प्रिपरेशन।
should I say that I am preparing for UPSC, or I should say that I am preparing for general examinations, any examination?
टू स्पेसिफाई इस यूपीएससी वुड बी वुड नॉट बी गुड फॉर यू।
Sir, I would say I am preparing for various
या
examination.
इट विल बी मच बेटर।
Yes.
Currently, I am preparing for government, various government, different government
Government
exams,
exam.
right?
Yes, sir.
Because
Because sir,
to
I have given state service also, and
yeah,
then
yeah, yeah.
UPSC
Because
also. So
because to specify UPSC examinations, they, that means you give a chance of to the interviewer to ask why you want to sit there here if you are already preparing for the UPSC examinations, right? So such kind of a
yes,
questions will be there arises, and you have to be clarify and justify unnecessarily. So just stated that you are preparing for different kinds of government examinations, right?
yes,
It's enough and
yes,
it's good for you.
yes, sir.
Okay.
So there is a gap of I completed my MTech in 2020, and so now there is a
2020.
gap of 3 years.
There is only
Yes,
1 year gap. It's
yes,
obvious.
sir.
After completing my PG, I was involved in preparing for the government exams, and government know very well there will be no regular form and there will be no regular vacancies. So what should I do?
So few years get wasted in Corona, and then
Also,
this.
yeah.
Yes,
So it's no issue. You completed your
sir.
master. Huh.
Sir, in area of interest, how many subjects would I state them?
Area
One or
of a
two?
subject, it will be much better you state one subject,
Okay.
or maximum one or two. Suppose you are confident on, suppose you are very confident on the fermentation technology and secondary immunology, right? Suppose, so you
Yes.
can
Yes.
you can state like that, sir. I am my favorite topic is fermentation technology, but I am also comfortable on immunology,
Yes.
लाइक दैट। इस तरीके से आप उसको बोल सकते हैं। सो उनको एक ऑप्शंस बन जाएगा कि अच्छा आपसे सवाल यहाँ यहाँ से कीजिए, और ये सवाल आपसे पूछा ही इसीलिए जाता है ताकि इंटरव्यूअर आपसे उस डोमेन पे सवाल बात कर सके,
Yes.
क्योंकि पहले से कुछ डिसाइड करके तो आते नहीं है कि क्या उनसे पूछना है। आपसे जब जैसे हमें थोड़ी पता है कि आप क्या हैं क्या नहीं, जब आपसे बात होना शुरू होगा देन आपसे हम सवाल करेंगे, फिर आप उसका आंसर करते हैं। तो
Yes, sir.
डेफिनेटली सेम थिंग विल बी देयर। और बेटर होगा कि आप उसको इस तरीके से बोले कि सर अब माई फेवरेट टॉपिक और माई फेवरेट सब्जेक्ट इज फर्मेंटेशन टेक्नोलॉजी, बट आई एम ऑल्सो इंटरेस्टेड एंड कंफर्टेबल इन इम्यूनोलॉजी। तो आप इस तरीके से बोलेंगे तो उनके लिए ठीक रहेगा।
Yes.
ठीक है? और थोड़ा ये है कि वैसे ठीक है, आपकी पकड़ आपके साइड पे ठीक है, बट मैं और चाहूँगा कि कुछ जो नई चीज़ें हो रही हैं, उसको थोड़ा गूगल कर लीजिए फर्मेंटेशन टेक्नोलॉजी में, और जिन-जिन पॉइंट को
Yes,
मैंने सजेस्ट किया, उनको थोड़ा-सा देख लीजिए, वो ज़्यादा अच्छा रहेगा। आपको
yes,
नर्वस
sir.
होने की ज़रूरत नहीं है, और इंटरव्यू में जैसे अभी आप यहाँ पे प्रेज़ेंट कर रहे थे, वैसे ही रहिएगा, कोई दिक्कत नहीं होगा। वहाँ पे आपके पास हेडफ़ोन नहीं होगा, तो और आप कॉन्फिडेंट फ़ील
Yes, sir.
करेंगे। और
Yes.
हाँ, बिल्कुल। और बाकी जो चीज़ें ऑब्वियस से होगा कि जो चीज़ें नहीं आएँगी उनको आप सिंपली डिनाई कर सकते हैं कि सॉरी सर, राइट नाउ आई एम नॉट रिकलेक्टिंग एनी कॉन्सेप्ट रिगार्डिंग दिस, एंड सो आई एम नॉट एबल टू स्पीक एनीथिंग एल्स।
Yes.
इस तरीके से आप डिनाई कर सकते हैं, कोई दिक्कत नहीं है। ओके, तो
Yes.
अल्टीमेटली ये है कि आपको अपने जो सब्जेक्ट है और अपना खास करके जो एरिया ऑफ इंटरेस्ट या फेवरेट टॉपिक है, उस पे थोड़ी सी अच्छी से पकड़ बना लेनी है, ताकि
So
एटलीस्ट
can I say, sir, I am confident in thermodynamics. So can I say that?
yeah, definitely. If you have keen interest in thermodynamics and if you have good idea about thermodynamics, then definitely go for it. And in fermentation technology, there must be a use of thermodynamics.
Yes. So there was one compulsory subject on thermodynamics in bichemical
Exactly. So
engineering.
definitely you should
And
say, okay, if
yes,
you are
yes,
more confident, if you have idea about the thermodynamics, then you say like that, I am confident in the fermentation technology and thermodynamics. So it will be very good.
yes, sir.
You are a MTech student, so definitely.
Yes, sir.
Okay. Anything else, Anand?
No, sir. I think if I will have any query, I will contact you, sir.
Definitely. How are you feeling right now?
So I'm feeling good because I was
Okay.
not prepared well, but it still it went well.
There was a network issue. That's why we not conducted properly. There was
Yes,
some mistakes in between. But meanwhile, there is no any issue. And I hope you are feeling more confident and comfort right now.
yes, sir.
Okay. And further, if you have any kind of a queries regarding the interview, then you are most welcome. You have my number. You can just message me and ask your problem. I will probably, whatever will be the possible suggestions for that questions, I will suggest you. Okay.
Yes, sir.
Just
So will I get this video?
yeah, definitely. I will share all the recorded. This video is going to be recorded, and I will share you to you.
Yeah. Thank you, sir.
Okay. Just keep
Yes,
this dress for the interview, and you are looking good, so there is no
yes, sir.
issue in your physical
Yes, sir.
appearances. Okay.
Thank you, sir. Thank you very
Okay.
much.
Thank you.
Thank you, sir.